In a cave tucked into the Altai Mountains of southern Siberia, a fragment of bone no larger than a thumbnail sits in a research lab, and inside its ancient structure lies a genetic signature that should not exist. The bone belonged to a 13-year-old girl who lived 90,000 years ago, and when scientists first sequenced her DNA in 2018, they found something that made them question everything they thought they knew about how ancient human species interacted with one another. Her mother was a Neanderthal, her father was a Denisovan, and she remains the only first-generation hybrid of two different human species ever discovered in the archaeological record. The girl, nicknamed Denny by researchers, represents a biological event so rare that despite decades of searching through thousands of ancient human remains, no other example has surfaced. This rarity poses a question that has haunted paleoanthropologists since her discovery, because genetic evidence shows that Neanderthals and Denisovans did interbreed with each other and with our own species, Homo sapiens, leaving traces of their DNA in people alive today. If interbreeding happened often enough to leave a genetic legacy in modern populations, why has only one first-generation hybrid ever been found? The answer, according to research published just weeks ago, may lie not in how often these ancient humans met and mated, but in what happened to their children afterward. Denisova Cave, where Denny's bone was found, has become one of the most important archaeological sites for understanding ancient human diversity. The cave sits in a remote region where the borders of Russia, Mongolia, China, and Kazakhstan nearly meet, and for tens of thousands of years, it served as a shelter for different human groups moving through the landscape. Archaeologists working there have uncovered evidence that Neanderthals, Denisovans, and even early Homo sapiens all used the cave at various times, sometimes overlapping in ways that suggest they were not just passing through separately, but encountering one another. The bone fragment that would later reveal Denny's identity was discovered in 2012, but it took years of painstaking genetic work to understand what it represented. When researchers extracted DNA from the bone, they expected to find genetic material from one species or the other, Neanderthal or Denisovan, because those were the two groups known to have lived in the region during that time period. What they found instead was a genome that showed roughly equal contributions from both species, which meant this was not someone with a distant ancestor from another group, but a person whose parents belonged to two different branches of the human family tree. The discovery was so unexpected that the research team ran the analysis multiple times to make sure there had been no contamination or error, because finding a first-generation hybrid was considered highly unlikely given how few such individuals had ever been identified in the fossil record. The significance of Denny's existence extends beyond the simple fact that her parents were from different species. Her genome also revealed that her Denisovan father had Neanderthal ancestry himself, meaning that interbreeding between these groups had happened in previous generations as well. This suggested that the boundaries between Neanderthals and Denisovans were not rigid, and that when these groups encountered one another, they did not treat each other as entirely separate or incompatible. The genetic data painted a picture of a world where different human lineages met, interacted, and sometimes formed relationships that produced offspring. Yet despite this apparent openness to interbreeding, the archaeological record has yielded only one confirmed first-generation hybrid. For years, researchers proposed various explanations for this paradox. Some suggested that the groups simply did not encounter each other very often, which would make hybrids rare by chance alone. Others argued that hybrid individuals might have been socially marginalized and therefore less likely to be buried in ways that would preserve their remains. A third possibility was that hybrids faced biological disadvantages that reduced their survival or reproductive success, but without direct evidence, these ideas remain speculative. Then, in October of this year, a new study offered a potential answer that ties together genetics, reproduction, and the fate of ancient human hybrids in a way that had not been fully explored before. The study, published in a leading scientific journal, examined genetic incompatibilities between Neanderthals and Homo sapiens, focusing specifically on genes involved in red blood cell function and immune response. Researchers found evidence of a slow-acting genetic mismatch that would not have caused immediate problems, but could have increased the risk of pregnancy complications in women who carried hybrid fetuses. The mismatch involves variations in genes that affect how a mother's immune system recognizes and tolerates a developing fetus, and when those genes come from two different human lineages that had been separated for hundreds of thousands of years, the result can be a heightened risk of miscarriage or stillbirth. This finding does not mean that all hybrid pregnancies would have failed, but it suggests that they faced higher risks than pregnancies within a single species. If a similar incompatibility existed between Neanderthals and Denisovans, which is likely given that they diverged from a common ancestor around 400,000 years ago, then many hybrid pregnancies may have ended before the child could be born or survived long enough to leave remains in the archaeological record. Deni, in this context, may represent not just a rare event, but a successful one, a hybrid who survived the genetic odds and lived to adolescence in a world where many others like her may not have. The implications of this research reach beyond the ancient past and touch on questions that matter today. The same genetic mechanisms that may have caused problems for ancient hybrids are still active in modern human populations, and researchers have noted that certain combinations of genes inherited from Neanderthals and Denisovans can affect immune function, disease susceptibility, and even pregnancy outcomes in people alive now. A small percentage of pregnancy losses in modern populations may be linked to genetic incompatibilities that echo the challenges faced by ancient human hybrids, which means that understanding Deni's story is not just about reconstructing the past, but about making sense of biological patterns that persist in the present. While the genetic incompatibility hypothesis offers a compelling explanation for why first-generation hybrids are so rare, it also raises new questions about how interbreeding shaped the evolution of our species. If hybrid offspring faced reproductive challenges, why did Neanderthal and Denisovan DNA persist in modern human populations at all? The answer may lie in the fact that not all hybrid individuals face the same level of disadvantage, and those who did survive could have carried advantageous genes that helped their descendants adapt to new environments. Recent research published in August of this year found that a gene called MUC19, inherited from Denisovans, played a role in helping indigenous peoples of the Americas survive in the New World by shaping their immune responses to unfamiliar pathogens. This suggests that even if many hybrids struggled, the ones who thrived could have left a disproportionate genetic legacy. The story of Deni has also been enriched by discoveries made in the past year that are filling in gaps in our understanding of what Denisovans looked like and where they lived. For most of the time since their discovery in 2010, Denisovans were known almost entirely from genetic data, with only a handful of bone fragments and teeth to suggest what they might have looked like. That changed in June of this year when researchers announced the identification of the first near-complete Denisovan skull, a massive cranium discovered in China that reveals a prominent brow ridge and a brain as large as that of modern humans and Neanderthals. The skull was identified not through DNA, which had degraded too much to be useful, but through ancient proteins and calcified dental plaque, which provided enough information to confirm its identity. This discovery, along with the identification of a Denisovan jawbone found off the coast of Taiwan and published in April, has expanded the known geographic range of Denisovans far beyond the single cave in Siberia where they were first identified. It is now clear that Denisovans were not a small, isolated population, but a widespread group that occupied diverse environments across Asia, from the high-altitude Tibetan Plateau to tropical islands in Southeast Asia. This geographic spread increases the likelihood that Denisovans encountered other human groups regularly, which makes the rarity of hybrids like Deni even more puzzling and reinforces the idea that biological factors, rather than simple lack of contact, played a role in limiting the number of successful hybrid offspring. Another recent line of research has taken a more experimental approach to understanding what Denisovan and Neanderthal genes actually did in living organisms. In October, scientists published results from a study in which they inserted Neanderthal and Denisovan gene variants into mice to see what physical effects they would produce. The mice developed larger heads, twisted ribs, and shortened spines, which suggests that these genes influenced skeletal development in ways that could have made hybrids physically distinct from their parents. While these results come from mice and cannot be directly applied to ancient humans, they offer a glimpse into how genetic differences between species might have manifested in visible traits, and they raise the possibility that hybrid individuals like Deni may have looked noticeably different from either Neanderthals or Denisovans. The question of what happened to other hybrids, if they existed, remains open. It is possible that many did not survive to adulthood, or that they were absorbed into one population or another and are difficult to identify in the fossil record because their remains are fragmentary or have not yet been found. It is also possible that some individuals who appear to be Neanderthals or Denisovans, based on physical features, actually had mixed ancestry that has not been detected because their DNA has not been sequenced. As more ancient genomes are analyzed, researchers may find additional examples of hybrids, though the fact that none have been identified so far despite extensive searching suggests that Denny's case will remain exceptional. What makes Denny's story particularly striking is not just the genetic data, but the human dimension it reveals. She was a child who grew up in a world where her mother and father came from lineages that had been separated for hundreds of thousands of years, yet they came together and produced a daughter who lived for at least 13 years. The circumstances of her life, how she was raised, whether she was accepted by one group or both, and what ultimately happened to her are questions that cannot be answered with the evidence currently available. What is clear is that her existence challenges simplistic narratives about ancient human behavior and shows that the boundaries between groups were more fluid than often assumed. The broader picture emerging from research on ancient human interbreeding is one of complexity and nuance. Neanderthals, Denisovans, and Homo sapiens were distinct enough to be recognized as separate lineages, yet similar enough to produce offspring when they encountered one another. The fact that these offspring sometimes face biological challenges does not mean that interbreeding was unnatural or doomed to fail, but rather that evolution had shaped each lineage in ways that made their genetics less than perfectly compatible. The persistence of Neanderthal and Denisovan DNA in modern humans shows that despite these challenges, some hybrid individuals not only survived but thrived, passing on genes that continue to influence human biology today. As researchers continue to study ancient genomes and uncover new fossils, the story of Denny and others like her will likely become richer and more detailed. Each new discovery adds another piece to the puzzle of how our species came to be, and how the complex interactions between different human groups shape the genetic diversity we see in populations around the world. The fact that Denny remains the only first-generation Neanderthal-Denisovan hybrid ever found may change as more sites are excavated and more bones are analyzed, or it may stand as a testament to just how rare such individuals were. Either way, her story serves as a reminder that human evolution was not a simple linear process, but a tangled web of encounters, exchanges, and genetic experiments that played out over hundreds of thousands of years. The genetic legacy of these ancient encounters is still with us, written into the DNA of billions of people, and the research being published today is helping to decode what that legacy means. From immune system variations that affect how we respond to diseases, to genes that influence our physical traits, and even our risk of certain health conditions, the echoes of ancient interbreeding are everywhere. Denny's existence, preserved in a tiny fragment of bone from a cave in Siberia, is a window into a time when the human family tree had more branches than it does now, and when the question of what it meant to be human had a more complicated answer than it does today.